Good afternoon, everyone. Is now audio clear?
Yes,
Yes, sir.
Yes.
Thanks. Yes, welcome back to the chapter on transcription. We were discussing about the sigma factor, and actually we are talking about prokaryotic transcription. We spoke about RNA polymerase. We spoke about the importance of sigma factor. We also spoke about the importance or the types of promoters. Promoter with -10 sequences, TAT, AAT, the consensus sequence. There could be variation in different genes and promoters. -35, it is TTG, ACA. This is for E. coli. Different species may have differences in the recognition sequences of -10, -35, and so on. Up element is the upstream regulatory element. And there are other types of promoters also, promoters like extended -10 or a promoter with discriminator region and so on. So the RNA polymerase has the several subunits, of which alpha subunit has C-terminal domain extender outwards, which interacts with up element. It's not necessary that all promoters have up element. That's one thing you need to keep in mind. And the broken arrow that is shown here indicates the +1. When you say +1, that is the first base added during transcription or transcribed. And we also spoke about sigma factor, which is the protein component that recognizes the regions -10 and -35. It can be divided into several subparts of the sigma factor, as in sigma part 2 interacts with -10, and sigma 4 part 4 it recognizes the -35 region. So sigma factor is the one that actually allows the RNA polymerase to interact with the promoter. Yes. And I was talking to you about there are different types of sigma factors. So bacteria has, as we will discuss, I think it might be premature to bring it now, but I would want you to have a better understanding at this stage. Say, for example, if there is a bacterial genome, okay, I'll put it like this, and this is the DNA. Assume it is chromosome. I'm writing two strands. Maybe I will erase that and say one strand. And I'm putting different types of genes around. Okay, so there are some, say I've drawn some genes. I will name them also. Gene A, B, C, D, E, and F. And say the intent of what I'm trying to say here is that bacteria are single-celled organisms. They live in the environment, maybe in the gut of other animals or in nature. That means one single cell has to withstand different types of stress. What is a stress? Anything that is not optimal is stress. Stress could be because of pH, it could be because of, say, salt concentration, it could be because of heat, it could be because of starvation, everything. So basically, one can say that bacteria in general are always under stress. So what is this related to in terms of transcription? I'm going to say that, say for example, in pH also you have two different types, right? Acid and alkaline conditions. Okay. Different bacteria have different optimal pH, for example, or salt concentrations, temperature, and starvation, ability to withstand starvation, and so on. So I'm just talking in terms of E. coli, Escherichia coli that's in our gut. And it can also live in the surroundings, in water bodies, and so on. if an E. coli, say for example, gene A is important for regulation during acid conditions, okay, and the gene E and F are for alkaline conditions. That means what I'm intending to say is they would produce a protein. That protein will help in mitigating alkaline conditions, right? Allowing it to survive during alkaline conditions. That is what I meant by it. And say B is for heat and D is also for heat. And say H and G are for starvation. I'm just making up a scenario. Okay. When do you want these genes to be produced? Do you want all these genes to be produced? Say, for example, if it is in optimal conditions, you don't want any of these genes expressed. You only want those genes, metabolic genes, and so on, to carry out glycolysis and produce more protein. So you would want ribosomal genes to be expressed and so on. Here I'm talking about stress proteins, just for understanding's sake. In the next slide, you will get to you'll understand what I meant. So if all of a sudden, if you take some of the genes like this, you will call them as housekeeping genes. They're always required for production. I mean, they are required to be produced so that metabolism and survival of the bacterium does happen. So for that, bacterium E. coli has something called as sigma 70. That is one type of sigma. If all of the genes that I have mentioned, if all of them had the same type of, or can be recognized by sigma 70, then all the genes would be expressed. And that's not important. That's not useful in optimal conditions. But if you have changed to conditions such as the temperature is because of heat or high temperature, then say we will just say it as high temperature. Then you want the expression of gene B and you want the expression of gene D. You do not want the expression of other genes, right? So what E. coli has in its genome is something called as sigma 32. That means if sigma 32 is present during heat shock, there is sigma 32 gene for sigma 32. So sigma 32 is produced, and sigma 32 will act on these particular genes, and then they will produce proteins required to help in tolerating the stress. So bacteria have different sigma factors, and each sigma factor is functional at certain conditions, specific conditions. And then the genes which have those promoters responsible for sigma, those typical sigma factors will help in mitigating the stress. Here are some of the sigma factors. RPOD, this is for E. coli, okay? This is the housekeeping sigma factor, or when all the conditions are good, or majority of the genes have sequence similarity. You can see -10 and -35. Most of the housekeeping genes and majority of the genes have sequences that are consensus sequences. At -10 it is TAT, AAT, and TTG, ACA. But then again you have RPOH gene which produces sigma 32, that is during heat shock. So those genes that produce proteins important for withstanding heat shock will have these sequences. At -10 it has something like this, and -35 it has something like this. So there are several others, nitrogen starvation and so on. What I want you to remember is there are several sigma factors, and these sigma factors recognize specific protein promoters. These promoters are present upstream of certain genes. And these genes produce proteins involved in stress, that specific, in mitigating that specific stress, right? If there is heat shock, sigma 32 is produced and it recognizes to sigma 32 specific promoter, which is usually upstream of genes that are involved, that can produce proteins that can help in mitigating the heat shock. Like that for cold shock and so many other things that. So while this is more for you, all you need to remember is know that there are multiple sigma factors. I had another picture. I think it is missing, but that's all right. I will show it somewhere else. So we also learned about the RNA polymerase, the channels. It does a lot of functions. It unwinds here, it rewinds here. It polymerizes the RNA, and it will exit the RNA through this channel. If you see this Q sigma 1, 3, 4, that is the sigma factor that we have been discussing. And we also spoke about something called as abortive initiation, where we meant that there is a RNA polymerase, it forms open complex, it forms closed complex by binding to the promoter. That then forms open complex where there is a transcription bubble, and then there is progress towards elongation. But elongation does not resume. So sometimes the RNA polymerase just recognizes or makes small fragments. It binds here, and this is assumed this is the promoter. It only travels only a few bases. At the end, resulting in several times it will do, only small fragments of RNA are formed. And that is abortive initiation. Initiation is not progressing towards elongation. Sorry. So one of the reasons could be that if you notice that sigma factor is here, is the sigma factor is obstructing the channel for RNA exit. So that could be one of the reasons why this abortive initiation may happen. Okay. And we also discussed about polymerization. So all of the polymerization is nucleic acid polymerization is very simple, or it is similar compared with in relation to DNA polymerization. All you differ here is you have R and TP, or you can just say as NTPs. NTP would be A, G, C, and uracil. And the difference between DNTP and RNTP, RNTP you have ribose and not deoxyribose. So similar to DNA polymerase, you have two metal ions, magnesium ions, which will stabilize the alpha, beta, and gamma phosphates of the incoming base in a particular orientation, so that the 3' OH will make a nucleophilic attack at the alpha phosphate, and this is broken, and now you have a phosphodiester bond formed. That is polymerization. And one other important thing about RNA polymerase is it does not require a primer, unlike DNA polymerase. So in today's class, what we will learn is more about elongation. So elongation is probably the simplest one. As I was describing about the polymerization, the RNA polymerase will just keep traveling in this direction, this way, and as it is moving, it will keep producing the RNA, right? There is no big deal about it. Only polymerization is happening, and you have to remember that it is now dissociated with sigma factor, okay? And so sigma factor is only important for, it is essential for loading of RNA polymerase at the promoter site, okay? Yes. So one important thing that we'll need to discuss in elongation is proofreading. That is ability to check one's own mistakes. I think that is something, if we look at several of the enzymes like DNA polymerase, RNA polymerase, and so on, they have a ability to correct themselves. We ourselves do not. We tend to, we have our biases there. We will not be able to recognize our own mistakes. Enzymes have this ability to proofread. One thing is, first of all, if you have a T, C, A, G, and if polymerization is happening in this direction, say this would be 3 prime, and this is 5 prime. So if RNA polymerase is being synthesized, U, A, G, and C. If it added a C, it doesn't add a C typically. It has mechanisms to recognize what is the base here and add appropriate base in that location. While it is also doing it at a very fast rate. It's not the same as DNA polymerase, but it is polymerizing very fast. But when it does, it sometimes may make a mistake. And because of that mistake, now GU is a mistake. And that is what is proofreading, its ability to correct it. It will remove that one base if it has made, and then it will redo the base pairing and polymerization. In this point, proofreading, there are two mechanisms. One is phosphorylated editing. This is exactly what I have described. I said there was a U added, then the base pairing is not appropriate, so RNA polymerase will kind of stop and then add the correct base opposite to the base, opposite to T. It's a simple back reaction, because when there is a mismatch, RNA polymerase cannot move or kind of stagnates in that location, and then that is how it corrects. Sometimes the next one is called as hydrolytic editing. I think we have a picture. We should be able to describe one of it. This is the backtracking one. Just move by -1 and then remove that part. Right? It has the RNA polymerase has moved one step forward, and then there is a cut. And then you have remade. In some cases, you may have, when there is a base that is made, if there was a mistake, and it has traveled a C, for example, and it may have moved further, but this is a mismatch. In some cases, what may happen is it will travel several times and then make a cut somewhere here. Okay, that is moving back, and it is almost the same, except that it's not just about one undoing the reaction. It is actually going back and cutting out this part and removing the error containing sequence. So there are sequence factors like GRE and NUS factors, which will stimulate the hydrolytic activity of the RNA polymerase. So the next step is terminator. If there is DNA and the RNA polymerase is moving on top of it, assume that this is the promoter and RNA polymerase has started moving and it is moving in this way. How long should it go? Say, for example, the open reading frame is only from here to here. That means the RNA required is from here to here at the most. What is the point of going to produce a very long RNA when the ORF is only this much that is required for production of proteins? All this part of the RNA is a waste. It's waste of energy and resources, because again you have to degrade. You have produced, you consumed energy for doing that, and again you have to degrade so much. So it's twice a loss. So through evolutionary processes, there has been some refinement about if this is a ORF, we only need this much and avoid wastage of resources, right? And nature will favor those reactions, those molecules, those activities, behaviors, and organisms that do not waste energy. Because the constraint is that, say, this is an organism, it gets some resources. And the resources are matter and energy. That is always a constant stringency. Matter, the environment does not supply them abundantly just like that forever. Maybe for brief moments. Nature may supply, but it's not going to last forever. Or at least, you can also think of it. If you provide some time, and over time this is going to reproduce. There are two problems. One is you're consuming this, the limited matter and energy, and you're increasing the number of consumers. So there is going to be stress on the organisms that are here. So those organisms that can efficiently utilize the limited resources will survive, and those that are just wasting a lot of energy are unlikely to survive. That's how evolution works, right? It's a dynamic thing. And that is how all these processes actually have come about. Evolution will favor all those that can reproduce and all those that can survive or exist, and then all those that can reproduce, right? And again, there is a competition between all those that are existing and reproducing, and that is natural selection. And how variation brings about differences in different organisms, and then the natural selection will pick the best fit for that contextual ecosystem. Okay, so because of this kind of phenomena, there should have been some sequences around here. Those sequences should somehow, the termination should have occurred here, right? And for that, genetic elements called as terminators are there. And there are two different types of termination processes that we will look into in equal. One of it is called as row independent, and the other one is called as row dependent. Row is a protein that you will see, and if row is involved, then you call it as row dependent. If row is not involved, then you call it as row independent. The row independent terminator, you'll also call it as intrinsic terminators. What do you mean by intrinsics? Intrinsic means it is within. What do you mean by within? Within the sequence itself, right? So see, for example, this is row independent terminators. It can form a hairpin loop. Upon production of RNA, it can form a hairpin loop like this. So this is where the RNA is there. Assume that this is where the ORF is here. Okay, so this has been the direction of transcription. And now at this end, when a hairpin loop is formed and RNA polymerase is something like this here, because of this hairpin loop, the RNA polymerase will stop polymerizing further. And also, if you notice that you have multiple U's here, poly U. Poly U, I mean, why not poly T or why not GC? GC has 3 hydrogen bonds, whereas AT has 2 hydrogen bonds. So the number of hydrogen bonds between the DNA, right, DNA template, and these will be lesser. And this hairpin loop will also stimulate the dissociation of RNA polymerase from the DNA. So these are called as row independent terminators because row is not involved. And you can also call it as intrinsic terminator. I like that word because because it will immediately suggest you that intrinsic, meaning that the sequence itself can act as a terminator of transcription. And the next one is, this is how it will happen. I forgot about this picture. Yeah, so you end up having a poly A tail and assume that this is the region of the sequence where it can form a hairpin loop like this. Once it forms, they dissociate. That is row independent terminators or intrinsic terminators. Row dependent terminators are something that is involving a protein. It is a protein that there are some sites called as RUT sites, row utilization sites. Okay, row and UT RUT sites. So let me try again. I'll try to better the picture. So assume that this is the open reading, the DNA template, and here is the transcription site, start site, and -10, -35 would be somewhere around here. And you have the start codon, or this is the open reading frame. Right? Downstream of the stop codon, you may have certain regions called as RUT sites. So upon transcription, it will the RNA will be generated something like this, and once it is here, the RUT site will now be transcribed, and the row protein will recognize and will bind to the DNA somewhere here, and it will stop. It binds to the RNA with its ATPase activity, and it will kind of stop the RNA or dissociate the RNA, the transcript from the RNA polymerase. Okay, so RNA polymerase is supposedly something like this, and the row factor will kind of will kind of dissociate the RNA from the RNA polymerase by using its ATPase activity. Okay, so the RUT sites are usually they are rich in C, and they are about 40 bases in length. Okay, so terminators are termination can be in two pathways. I'll just quickly summarize: row independent and row dependent. Which one do you think is more logical? I would go with the row independent. It's all right to have small opinions built, so that it's important to have opinions, because every opinion is likely to have a reason. And when you have a reason, that is when you are actually, your capacity of reasoning is intelligence, in one form at least. And the key, however, is that you should be able to change your opinions based on what is the evidence and the information, increase in information, or authenticity of the information. Why I like row independent terminators is because it is intrinsic. That means it doesn't require any external, some other factor to come and inhibit or terminate the transcription here. But somehow evolution has come up with two, and there could be some benefit of it, right? But this is kind of expensive. Probably it is likely that the row independent terminator is not so efficient or as efficient as the row dependent ones. But evolution in general will bring about anything that works. It's not about the best thing that works, right? It's not always the case. It gives anything that works. Anyway, let's move on. So we'll quickly have a discussion on the transcription of eukaryotes, and I would want you to think about it again. We started this slide, our transcription lesson, with this slide. because I wanted you to understand where things happen. Replication happens in nucleus, so does transcription also, right? And upon transcription, there is a step, and it is called as RNA splicing or RNA processing. Processing is a better word because it's not just about removing the introns. There are three key factors that we would have to know. One is the cap, second is removal of introns, and the third one is, let me try, cap. First one is, the second is to remove the introns, and the third is to have a poly-A tail. This is true for majority of the eukaryotic organisms, right? So why we will have a lesson on RNA processing, that will be the next one, but we should also know how it is related, right? Cap is, you call it as G cap, and splicing is removing of introns and joining of exons, right? And then poly-A tail and G cap both are essential for translation. So the mRNAs are recognized by the presence of G cap and poly-A. There are two things important. One is recognition by the translation elements. Second thing is they are also important for stability of the mRNA. Okay. And I hope you know that it's important to remove introns, otherwise open reading frames are not complete. Okay. So now we will have a brief discussion on the different types of different aspects of eukaryotic transcription. There are three different RNA polymerases. In prokaryotes, there is only one RNA polymerase gene or complex. In eukaryotes, there are three different ones. You call them as RNA pol 1, pol 2, and RNA pol 3. Most of the mRNAs are synthesized by RNA polymerase 2. The rRNAs are synthesized by RNA pol 1. So it must be more in the nucleolus. Because I described to you that the ribosomes, the production of rRNA and assembly of ribosomes happens in the nucleolus. So RNA polymerase is likely to be in the nucleolus. And then there is RNA polymerase 3, which is mostly about tRNA, generation of tRNAs and other small RNAs. Unlike prokaryotic transcription, eukaryotic transcription is complex. It is, there are too many factors and so on. And we will know some of them, and some we will not discuss as much. But try to know that it is complex and there are many many factors that are involved. Majority of them that are common in transcription of majority of these are called as GTFs, general transcription factors. Okay, but when we within the cell, there are many other components which are something like mediator complex. If you remember properly, when we were learning about the organization of the genome, we learned that the DNA is bound around nucleosomes, and these will form a 30 nanometer fiber. And then you have higher degree of organization. And we also learned a little bit about modeling the nuclear organization so that the genes and the regulatory elements are exposed so that transcriptional or protein specific for transcription can bind or find access to the genes or regulatory regions and initiate transcription. So we need several things. That is what is about it. If you want, you can quickly have a look at that chapter and come back. So, but we need mediator complex. There are several proteins that are DNA binding regulatory proteins, and there are also chromatin modifying enzymes. I hope you remember something like chromodomains and bromodomains. Enzymes containing chromodomains and bromodomains. So the first thing is the promoters. We will We will discuss about it. There is a core promoter, which is the minimal set of sequence elements required. That's about 40 bases. Then you have more promoters, that is more elements, that is upstream and downstream. This assume as the core promoter, and you may have several factors on either of side. This will be the downstream if I'm talking this, if this is the reference point, and this is the upstream, right? We will quickly discuss what is with the type of sequences that are present in a promoter typical of RNA polymerase 2. So this is the first one. The broken arrow is the +1. That is the first base that is where transcription is initiated. And that region is called as the initiator region. And I hope you remember we have a sequence in prokaryotes. We had a sequence like -10, which had a sequence TAT, AAT, right? Very similarly, you have a sequence here, which is not -10, of course, between -26 and -31. You have a Tata box. The sequence also is matching, if you can see, TAT, and then you have difference TAA. Here it could be, in this position, it could be ART, A and ART. Okay? So let me erase a few things so that it will be clear. So this is called as the Tata box, and that is the referred as TATA. Initiator region, that is where the transcription usually starts. And there are several other things that you will remember. Downstream promoter elements, where is TP? This is the downstream promoter elements. You have BRE, that is, BRE is, okay, let me tell you this. All the names that are written here are transcription factors. Okay? You have TF2, TF is for transcription factor. Okay? And then there are several factors, like here it is called transcription factor 2B. Because B is binding here, this region is called as B recognition element, or TF2B recognition element. Tata box is something like -10, so you'll remember it. Here it is TBP, Tata box binding protein. Okay. So this is TF2D. TF2D recognizes several regions. That is the it is initiator, DCE1, DCE2, and also these DPE and so on. So with different affinities or different parts of the TF2D can be recognizing different regions. Okay, I hope you don't have to mug up everything. You don't have to mug up anything of it, actually. But try to know the names of some transcription factors, which we will learn when we are learning the initiation of the transcription in the coming slides or in the next class. Okay, so different regions have different sequences, and it's not necessary that all the promoters have all these elements. There could be more or less of these. Okay. So downstream core element. So DC1, DC2, and DC3. They are downstream core elements. And downstream promoter element is here. This BRE I have described. Tata box is described, and so on. Yes. So we'll go through this. I think I should have used the PowerPoint. In PowerPoint, I would have done that. I didn't check this probably. So just give me a minute. I'll just get back. View. I'll take this and I will stop. Okay. Yes. So here we have the first protein that recognizes is along with TF2D, TVP, that is Tata box binding protein, right? This binds here. It's almost similar to that of sigma factor we learned in prokaryotic transcription. Then transcription factor 2A and 2B. I think I have put this. So once the binding of TF2B, Tata binding protein and TF2B at the Tata box, and so there is a distortion of the DNA. And that upon distortion, the recruitment of TF2A and TF2B are recruited to the promoter. The TF2B has a function in recruiting the RNA polymerase. RNA polymerase is bound, RNA polymerase 2, we are talking about 2, is bound by TF2F. And then you have the loading of RNA polymerase to the promoter, something like this. So we have different functions. TF2F will interact with TF2B, and that is how TF2B is interacting with this one, and TF2B will interact with TF2F, and which is in binding, which is bound to RNA polymerase. So you have RNA polymerase in the promoter bound to the DNA. That is called as the pre-initiation complex. And then once the pre-initiation complex is formed, you have the recruitment of TF2E and 2H, and that is the 2H, H for helicase is what you can remember, which will unwind the DNA. Here is the transcription bubble that has opened up. Okay, and then it also phosphorylates RNA polymerase on the tails like this, and which will allow something called as promoter escape. Why do you call it as escape? RNA polymerase is recruited to the promoter. How is it recruited? Because it is recruited by binding to the several factors that are associated with the promoter. That means if it has to get rid of that interactions with the other proteins and move on, right? It has to escape those the interactions with these proteins. Only then it can move forward. So that's why you call it as a promoter escape. It is something very much similar to that of what would have happened with abortive initiation in prokaryotes. So this is a good page to remember how it's a good page for notes, right? It is from Leninger, initiator region, and you have the Tata box, and they just simply put TBP, TF2B, TF2F, along with polymerase 2 and 2E and 2H, which I said has the helicase activity. If you are going to prepare a notes, this is a good picture for a notes, right? And then of course you will have promoter escape, and then there is a closed complex, and TF2H using its helicase activity will make it as open complex, and then there are phosphorylation of Pol2 and initiation of promoter escape, and then RNA polymerase will move forward transcribing, and then you will have several of these proteins removed or they dissociate from the RNA polymerase, which will get recruited for a new round of initiation. Right? That is how transcription happens in prokaryotes. So I'm not going to say much about it, or I will repeat this in the next class as well. Just that, so that you will be able to retain it for longer time. There are several parts, several sequences within DNA that influence how much a gene should express. As you recall, as you know it as actually, that prokaryotes are much simpler and eukaryotes are disproportionately complex. And then that complexity comes from so many genes influencing every promoter. This is a promoter, there is an influence. The more number of things it requires, more factors involved, more complexity, from which more delicate and fine-tuning can happen. When I say more factors, it could involve several enhancers to which activators bind, and there are what we spoke of is just this much part in the previous slide, but that's not about it. We have several other proteins like complexes of proteins like mediator complex, chromatin remodelers. You have HAT, if you can recollect. It is histone acetyl transferase. I hope you remember HAT, histone methyl transferases. We also learned about HDAC, that is histone deacetylases. There are several modifications that could happen on the histone pairs, right? Please go back to this chapter and come for the next class, because that is what we will discuss in the next class when we get back. CTD, C-terminal domain. So I'm not going to describe it. I'm just trying to put some words because we already learned so many words today, or terminology or glossary. I just want vocabulary. So I just give a few more. Mediator complex, the one that you're looking at here, is a large complex of several proteins. It is interacting with the C-terminal domain of RNA polymerase. C-terminal domain of RNA polymerase is very important for what will happen in the future during the course of this transcription. Okay. And then there are several transcription regulatory proteins like activators. These are activators. There are also repressors. Okay, when we study about regulation, gene regulation, these all things will come in handy. So try to go back to the lesson on genome organization. I hope you can read the textbook. At least now you are in a good position to understand the book. Try to read the textbook and also follow this chapter very well, so that when we get to the gene regulation, we will feel comfortable. Majority of gene regulation happens at the level of transcription. So one must be very clear about what all can influence transcription, and especially in eukaryotes, the regulation of transcription is much more complex because there are so many things that are involved that can influence the promoter or the protein recruitment at the promoter. Okay, I will stop here, and what I would want is that. Okay, I'll take up in the next class, these parts in the next class. Why I'm showing this slide is that I have seen some of your assignments, and I think many of you have done a good job, considering that we haven't read about it and so on. I hope your knowledge from Biology for Engineers is helping you. I'm assuming that. For assignment 3, I will just turn off the recording.